All right, everyone, let's get started. So last time we talked about sequence alignment and mapping, and basically how that's the process of taking a FASTQ file, which is the raw sequencing reads off of a DNA sequencer, and finding their home in a reference genome or a reference transcriptome. And then once all the candidate homes for each read have been found, there's this nucleotide by nucleotide alignment that typically is done with the Smith-Waterman dynamic programming algorithm. What we're going to talk about today is some of the software that's currently used to do that alignment and the formats that those aligners produce for viewing alignment data and things like IGV, for calling genetic variants with things like GATK, if you've heard of that, or for looking for differential expression or ChIP-seq peaks with tools like MAX or I'm losing the name of the differential expression, but DE-seq. So this format is a bit hairy. It's even more complicated than any of the other ones we've looked at. We're going to try and go into some of the details today. There are some details in it that you really don't need to care too much about, so I'm going to try to cover the things that are most relevant. It's a pretty complicated format, but by the end of today's lecture, hopefully you have a good sense of the key elements in that format and how we use it. Okay, so to get started, we're first going to introduce two different Unix tricks. Most of the commands that you've run so far finish fairly quickly. If you do ls or head on a file, you basically type that command, hit enter, and it finishes the execution and gives you your prompt back. But as we're going to find, if you haven't found already, maybe even in homework 3, some of these commands can take a while. If the files are really, really big, that is, they have, say, billions of lines in them, in the case of a FASTQ file from a full whole genome sequencing run, it might take sometimes hours for those commands to finish. And the way Unix works, if that command is running for an hour, basically you lose control of your prompt. So it's essentially like the oldest versions of the iPhone. You'd open up app, you couldn't do anything else. You only had to be in that app. You could only be in that app. You couldn't switch. You couldn't multitask. So there's a trick in Unix where you can use, if you put at the end of any command an ampersand, it actually will put that work in so-called in the background. It'll launch that execution. It'll find a CPU that's available to do that work, but it'll immediately give you your prompt back. When that work is finished, you'll get a message on your screen that says, job essentially finished. And you can send lots of commands. You could type a command, ampersand, hit enter. Type another command, ampersand, enter, and launch a whole bunch of jobs behind the scenes on your computer. Now that will work up to the point where there's not enough CPUs left available on your computer to do that work, and those other jobs will just sit there and wait. So just as an example of this, I've got two FASTQ files that I'll put in your directories as well. Well, let me just show you. The wireless is really slow today, so bear with me. So I've got a file called 1.fastq and 2.fastq. This is from a paired end sequencing run. So the standard for a paired end sequencing run, where we're sequencing the 5' end, both 5' ends from each molecule. We end up getting two FASTQ files, one for all the 5' ends and one for the other 5' ends for all those clusters on an Illumina run. So if I look at the first few lines of this file and the first few lines of this file, one of the things we should see, so the first few lines of this, the first sequence in this file has this ID, SRR06234.1. And okay, the first ID for this file is the same as the first for the second. So this is one 5' end for the same molecule. This is the same 5', the other 5' end for the same molecule, and so on and so forth. So the ordering of these two files should be the same, the 5' ends from the same molecules over and over again. All right, so I just took the first 10,000 lines from a file I found, two files I found on the internet. But you know, if we wanted to sort these two files, this is painfully slow. Sorry. Wow. Okay, thank you. I'm not patient. Okay, so that's sorting and sorting and sorting, spitting out all the results. Okay, so these are relatively small files, just 10,000 lines, but imagine these had billions. That took a few seconds. For billions, this could take up to an hour. So if I just do this, maybe I want to write it to a file called sorted. If I put it in the background, and as soon as it responds, I get my prompt back, and it gives me a job ID right here. This is the name of the process that's running on the computer. Okay, so I have my computer, and then it interjects. It says, oh, it's done, and gives me my prompt back. So it reminds you of the command that you ran maybe 15 minutes ago, maybe even an hour ago, and tells you that's done. So now I have this new file in there that I can look at. But the basic point is, with the ampersand, you regain control of your prompt, so you can do multitasking. Now, there's another command called nohup, which stands for no hangup, that you can use in conjunction with an ampersand for a slightly different problem. Let's imagine you're going to trivia night with lab mates, and it's 5:30 right now, and you want to launch a job, and you want to get out of there and go meet your friends for trivia. If you shut down your computer, if you shut down your laptop, and you have a connection to Malibu, that job, any job that's running, will terminate because you've lost your connection to Malibu. You'd like a scenario where you could actually just like fire off some work, close down your laptop, go answer some trivia questions, win, come back in the morning, and the job kept running overnight and things were good. That's what nohup allows you to do. So basically you can submit a job, close down your laptop, and it allows this job to run in the background even when your connection has terminated. There are other alternatives to this. There's something called screen, where you can actually launch sort of sub-screens in your terminal to do this type of thing. The reason I introduced nohup is that not all computers allow screen. Not all systems enable screen. For instance, King's Peak doesn't allow you to use screen, or it may now. It didn't when I first got here. So nohup is sort of a general platform independent approach to just letting stuff run overnight. Okay, any questions about this? Yeah.
So for nohup, is the ampersand at the end necessary, or will that just, in case you want your prompt back?
It is, yeah. Yep. Okay, so now on to the meat of the lecture today. There's a lot of different sequence alignment tools out there, and they're broadly broken up into two different categories. Sequence alignment algorithms for DNA sequences and sequence alignment algorithms for RNA sequences. Now that's a bit of a misnomer because actually with Illumina, we don't actually sequence RNA. We take RNA and convert it to cDNA first. But these aligners actually are customized to know that when you have cDNA, it's usually from a mature RNA that has splicing junctions removed. So you've got exons abutting the next exons and so on. So when you align those sequences to the reference genome, it has to be able to tolerate really big gaps in the alignment, which actually reflect the introns that are in the reference genome. So we walked through the Smith-Waterman approach last time, And you can think about those introns as just really big gaps that go off the diagonal in that process. So you could imagine that that's atypical because Illumina sequencing technologies don't make insertion deletion errors very much. So there's kind of specialized logic in these tools to account for, potentially like tens of kilobases of gaps introduced by introns when you align cDNAs back to the reference genome. Probably the most widely used aligner out there today is BWA-MEM for Illumina sequencing. It's really quite good. It's insanely fast. It gives really accurate alignments and has been tested over and over again. STAR, actually, I'm not an RNA-seq expert. I think STAR is one of the more widely used aligners out there. If you know of other ones that people use, let me know. This list is probably a couple years outdated, but it gives you a sense that most of these tools, thankfully, are open source, and there's lots of different options to use. And I think depending upon the type of experimentation you're doing, you might want to read prior papers and look at the methods section to see what aligners they chose to use and perhaps why. Okay, so BWA-MEM is a kind of special paper in the sense that this thing has probably been cited 10,000 times, but it's actually never been published. The story is kind of interesting. If I remember correctly, Hung Lee, who's the author of this, sent it to Bioinformatics as a short 2-page paper, and reviewers basically said it's not innovative enough, reject. So he just put it up on this thing called arXiv, which is typically used in the computer science, physics, and math disciplines, and said, "Screw it, I'm not gonna resubmit. Just gonna put it up." And people just cite the arXiv paper because it's so incredibly good. This is a so-called responder operator characteristic curve, which is a mouthful, but it's basically showing the trade-off between false alignments on the x-axis and true alignments, accurate alignments. So an ideal aligner would have a shape that goes like this, like on this rectangle. And basically, each of the points on these lines, the lines represent different aligners, and each of these points represent different parameter settings for each of these aligners, and trying to measure the trade-off between false positive alignments and true positive alignments. So one thing you can imagine that harkens back to the previous lecture is if we're using a hash-based aligner, the K size or the hash size has some control over the accuracy of the loci that are chosen for each read to potentially align them to the reference genome. So if K equal 1, for instance, it's not going to be a very good, probably not a very good aligner. But if K, as K increases, maybe you get more and more accurate alignments. And so what this, in summary, what this is showing, BWA-MEM is this red line, and it's second best. The shape of this curve is second best to this aligner called Novo Align that I used a lot during my postdoc. It's just really slow and it's commercial. So what BWA-MEM is showing is that it got kind of similar performance to Novo Align in 497 seconds versus Novo Align took maybe 5 times that time. Novo Align is a really good aligner, but it's actually fairly expensive, so BWA-MEM has sort of taken over. All right. Right. So BWA-MEM, how does this work? Well, it takes 2 inputs. What do you think the inputs are? What is it trying to align? Yeah, it's taking FASTQ files. If it's single-end sequencing, you give it 1 FASTQ file. If it's paired-end sequencing, you give it 2 FASTQ files. And then the other piece of input is the FASTA file. And the output is this SAM format, which is a hideous mess, and I've intentionally made it that way. We'll talk about the details in a minute. So it is the input. One of the inputs is a reference genome in FASTA format, but not quite. It requires you to so-called index that FASTA file, the reference genome. So this is GRCH38, and the acronym stands for Genome Reference Consortium Human Build 38 .fasta is my convention to say that it's a FASTA file. Okay. So we have to run, before we can align sequences to a reference genome, we have to first index that reference genome. And that is essentially BWA building the equivalent of that hash table that we walked through on Tuesday. It's doing it with a specialized thing called the Burrows-Wheeler transform. The B and W in the name BWA stands for Burrows-Wheeler. We're not going to get into the Burrows-Wheeler transform, but if you're interested in how that works, it's really, really clever. There's a nice couple of lectures from a guy named Ben Langmead that you can Google. It's a pretty amazing approach, but it's sort of outside of the scope of this course. So once we've indexed that reference genome, and we've got essentially a lookup table for where each of the different k-mers in the genome exist, then we can run BWA mem, which is the name of the aligner. I'll skip this for a second. We have to give it the reference genome. So what happens when you index this reference genome? Behind the scenes, it actually creates like 4 or 5 additional files that represents that index. And those additional files get extensions after this file. So maybe the first file might be grch.38.fa.bwt.sai. whatever. And so if you just give it the name of the root name of the FASTA file that you did index, that tells it how to find all the other index files that it expects to see. Okay? So we give it the name of the FASTA file, which allows it to find the FASTA file and its indices. And then we give it, in this case, we're aligning paired end sequences, so we give end1 followed by end2 FASTQ files. And what it outputs, if this is whole genome sequencing, 16 hours later, what you'll get is a SAM file that represents, for all of the reads that were in those 2 FASTQ files, the locations in the genome to which they were aligned. Okay? So these 2 steps, you index a genome once, so someone in your lab can do that for whatever organism you're working with, and you never have to do that again. This step is done for every data set that you ever produce. Every sequencing run, you have to align it to the relevant reference genome. So you might have indices for the mouse genome, for Drosophila, different versions of the human genome, whatever. Okay? So this raises the question, where do we actually get FASTA files for reference genomes? And this is a common question. This course is sort of taking you through how you use tools and how you work with different formats, but oftentimes when you leave a course like this, you kind of like, okay, I know what to do, but I actually don't know how to get the data that's relevant. So actually that's something that Mike Cormier in my lab is working on, is ways to quickly access data sets like this. But I'm going to show you just sort of the old school way. One way would be to literally just type grch38.fa into Google and hit enter. And one of the first hits that you'll find is this page from the UCSC Genome Browser. How many of you have ever used the UCSC Genome Browser? Okay, great. So you're familiar. It's a really amazing website. I mean, the graphics are pretty outdated. It looks kind of ancient, but in terms of content and what you can do with it, it's pretty incredible. It is not only a genome browser where you can actually look at gene models and type in random coordinates or random gene names and see annotations for whatever genome you like to work with, but what's also really cool about the UCSC Genome Browser, and also Ensemble in the UK, is that they actually provide all the data that they render on their website to you through FTP sites, so you can download it and use that data for your own research. So if I go to this link, I see this web page, and it's organized. There's a quick link for human. It's a bit human centric, but then you can drill down into other mammals. I don't know what a bush baby is, but maybe we could go get the reference genome for bush baby or ferret or gibbon, Craig Venter, if we wanted. Hedgehog, I don't know, look through this, see what you think might be cool. Pika, oh, that's pretty cool. So if we go back, oh, I blew it. If we go to human, you can see that it's organized by builds. So what I was using was build 38 of the human genome. Here's a problem in human genomics and really genomics for every organism. There's often two different naming schemes for the same kind of files. So the Genome Reference Consortium, or the GRC, refers to this as human build 38. In UCSC parlance, it's HG38. Okay, so these terms are used sort of interchangeably, which is it's a standard source of confusion. If you go back to 2009, the previous build was called by GRC H37, but for reasons that are unknown to me, UCSC calls it HG19. And then they agreed to sync things up, and now we don't have HG20 through 37. We jumped to HG38. Okay, bit of an aside, but we can click on genome sequence files. Click on standard genome sequence files. And once these bits make it across the internet, let's see. We scroll way down. This is like a big README telling you all the contents that are in there. You'll see one right here, which is this file called hg38.fa.gz. So this is the soft masked, I'll talk about that in a second, assembly sequence in one file. So it's basically a giant FASTA file with chromosome, that greater than sign to represent the first chromosome, CHR1, then the 250 million nucleotide sequence for chromosome 1, then chromosome 2, chromosome 3, all in one gigantic text file. Okay? All right, so that's how you know if you go back to your own lab and want to do this type of thing, you would just go here and find the right file. Here it is, hg38.gz. I could right click, copy link address, and then you've got something where you can type wget, hit enter, and download it. Okay? Right, so I've done that. We'll get to that later, but in the future you'll almost certainly have to do something like that. Right. So, jumping back, the output of BWMM is this SAM format. So what's SAM format? This was defined in an application note in, I can't read that, 2009. It was, you'll recognize a few names here. Hung Lee is also the developer of BWMM. Another author is the 1,000 Genomes Project. And my PhD mentor and our colleague upstairs was also an author on this paper. So a couple of things about this. Prior to 2009, so around 2007, 2007 I think, the 1,000 Genomes Project was kicked off. And it was hugely ambitious. Okay, we're going to use this cool new Illumina sequencing technology to sequence 1,000 genomes. Actually, they ended up sequencing 2,504. But the idea was to use that whole genome sequencing data from all these individuals to characterize the vast majority of relatively common genetic variation that exists across humans throughout the world. Prior to the launch of the 1,000 Genomes Project, there really weren't any standards for representing alignment data. So one of the first couple of years of the 1,000 Genomes Project was trying to figure out which aligners work best for aligning these short reads to enable us to find genetic variation. Which variant callers work best. So there's a lot of like so-called bake-offs, like competitions to figure out what the best methods were, refining methodology. And it very quickly became an absolute nightmare to be able to compare the output of all these different aligners like NovoAlign, and prior to BWMM there was one called MAQ or MAC. There was one developed in Gabor's lab called Mosaic. They're all sort of competing one another. So this group got together at a 1,000 Genomes meeting. I think this was in Philadelphia, and sat down in a conference room and said, forget all that, let's come up with a standard. Everyone conform to this standard. It'll make our job of comparing the performance of these different things better. That standard became the Sequence Alignment and Mapping Format, or SAM format. It's a text-based standard, and it really is a standard. It is used today. It's been tweaked a little bit since that first definition, but only moderately so. They pretty much nailed it the first time. It has some idiosyncrasies. It's not perfect, but it's a standard, and standards in science are really, really useful because it allows direct comparisons. Okay? SAM format is a text file. Each line in that text file has 11 columns. Okay? We're going to walk through what those 11 columns are and walk through some examples of SAM files. Okay? I talked about this. 1,000 Genome standardizes it, but the strengths of it, of the SAM and BAM formats, I'll talk about BAM in a second, is that it's relatively simple to produce. There's a lot of good toolkits. We'll talk about something called Samtools today for working with these formats. And there's clever ways for compressing them. That's what BAM format is essentially, in something newer called CRAM format. And you can index them. We talked about indexing FASTA files. You can also index these files so that if you wanted to, anyone have a favorite gene that they want to name? No one? I do. ATM is my favorite gene. So if you go to the UCSC genome browser, IGV, and type in ATM, it can quickly jump to the right location in that big SAM file and just display all the alignments in that file that are relevant to that particular gene without having to work with the potentially billions of other alignments that are scattered throughout the genome. So it allows you to like quickly jump to loci that you're interested in without waiting hours. So it's really quite nice. Okay, so don't look ahead, please. Let's think through this. What do we really want to know for sequence alignments? What are the core pieces of information that we might want to know? A FASTQ file, those are completely anonymous sequences that are just random shattered pieces of DNA from a million cells potentially that we fed into the DNA sequencer. When we align those sequences, what do we want to know for each sequence? Yeah.
Where in the reference genome it matches.
Where? Yeah. So, for instance, chromosome or position. Yeah.
The edit distances for each.
Right. How many differences there were between the sequence and the reference genome? Yeah.
The confidence you have that sequence is actually aligned there.
Exactly. Another one is strand. Did it align to the reference genome strand or the reverse complement? All right. Great. We might also want to know what the sequence was, the actual sequence too, right? So you really touched upon sort of the core components of the SAM format. And I also lied to you a little bit. There's 11 core columns, but there's this 12th column, which is a series of additional tags, which really are sort of supplementary information that can be used by things like variant callers and other information and other tools. But let's walk through this bit by bit. Let me try and turn my pointer on here. Okay, so the first column in the SAM format is something called QName or query name. This is literally just the read ID from the FASTQ file propagated as is to the first column. And so it's for bookkeeping. There's a system of record that connects every input sequence in the FASTQ file to an entry in a SAM or BAM file. Okay. There's this thing called the flag. This is going to require a lot more discussion, but it's a number that represents a lot of attributes about the alignment. One example is the strand to which the sequence aligned. We'll get more into that. As Sarah mentioned, we might want to know the location. So column 3 tells us what chromosome each sequence aligned to. In this case, this sequence HWA blah 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 aligned to chromosome 1, and it aligns starting at position 8,724,005. Okay. It aligned to that location fairly confidently. The mapping quality, as we talked about last time, is 60. So that means that, let's say BWA aligned this, it thinks that there's a 1 in a million chance that this location, chromosome 1, 8724005, is not the right location. There's a Cigar string that gets at Simone's idea, which tells us the sort of the nucleotide by nucleotide alignment of the sequence to the reference genome. We'll go into that in the gory detail. You might hate me for it, but we're going to do it. Okay, so let's imagine it's a paired end. This fragment at cluster HWI, ST156, etc. at this XY coordinate on that tile of that lane is a paired end sequence. This is saying that one 5' end of that sequence aligned to chrome 1 8724005, and the other 5' end aligned at chromosome 1 8724505. So roughly, how big is this fragment? Yeah, about 500 base pairs, right? And there's another, the next column kind of gets at that. It's about 500. It says 562. Why is it not just 505 minus 005? This coordinate is actually talking about the leftmost start of the alignment, so it could continue on the length of a given sequencing read. Okay. This next column, the 10th column, actually gives us the sequence of the query. So the query in this case is the 5' end that aligned here. This is just telling you the location of the mate, but doesn't tell you the nucleotide sequence of the mate. It just tells you the nucleotide sequence of this end. Okay? This comes directly from the FASTQ file as well. So the sequence ID comes right from FASTQ, the sequence comes from the FASTQ, and guess what? The quality string comes right from FASTQ as well. It's literally just carried over. So there's some redundancy here. I mean, essentially, these FASTQ files are going to be ginormous, and then we're restoring a lot of this data in the SAM and BAM format. You can imagine a scheme where the SAM file just stores all these positions and then gives you sort of references to the FASTQ file so you can look up the sequence. That's not what was done, for better or worse. So we restore all this data. And then these optional tags tell us things like the edit distance. I think that's what XA is. This is saying there's an edit distance of 2, and this is giving some more detailed information about the alignment that can be used by things like GATK. All right. Any questions about the basic structure of the file at this point? Yeah.
Sorry, can you go over again with the insert size? How it's 562?
Yeah, I wish I had. The unfortunate thing about this room is there's no whiteboard. There is? Oh my gosh, magic.
Put that up.
All right, so let's imagine this is the reference genome, and you've got one sequence, one end of the pair that aligns on the same strand as the reference genome. There's this unsequenced portion, and then the other end is going to align on the other strand. Okay, so this is so-called positive strand. This is negative strand. I forget what the leading digits were, but this is position 005, if I remember right. And this would be position 505. So this, from here to here, is 500 nucleotides. But what it's saying is this is an additional 62. So the insert size, so there's confusion. This is one of the things that has evolved in the format. So originally, that value represented from here to here. Then it represented from here to here. And in some aligners, it represents from here to here. So you have to know it can be used for different things. And that's where the specification of the SAM format has changed, like what that actually is representing. Does that make sense, though? Okay. Okay, so a concrete example. It's probably not all that visible in the back. Can you make it? Well, you have it on your screen, hopefully. So here is I ran Samtools, which we have just alluded to at this point, this command called view on a BAM file to look at the first record, this head-1. We're just looking at one record, the first record in this BAM file, which is just a fancy equivalent of the SAM file. And what you can see is the first column is the sequence ID. Then we've got this flag, which I've sort of just barely touched upon. Then we got the chromosome, position, the mapping quality. In this case is 0, so it's basically saying, yeah, I put it here, but could be anywhere. This cigar string, 51M means there's 51 nucleotides that match the reference genome. And it's not quite an exact match. We'll talk about that in a second. But that is basically saying it's a pretty close alignment between the sequence that's in column 10 and the reference genome. I forget what this is. Oh, that's basically saying the mate for this sequence aligns to the same chromosome as the query. So it's given an equal sign, saying basically that the chromosome for this is equal to that chromosome. Its mate aligns to 50214. The insert size is 132. Sequence right there, quality string, and then all these extra columns here are these optional tags, which we don't need to worry too much about. Okay, questions? Yeah.
So you said there's 51 matches.
Yep.
But is that pretty good, or like why is the score so low?
Yeah, so that's a great question. So there's 51 matches, so you might think, well, that should be a very high mapping quality. But those matches, that's the alignment. That's the nucleotide by nucleotide alignment. Imagine that 51 base pair part of the reference genome to which this read aligns occurs 1,000 times in the genome. That's why it's going to get a low mapping quality. I put it here, but it could go to any one of these 999 other places equally well. So it's reflecting the uncertainty in the actual location rather than the alignment itself.
So like places in the genome that are repetitive, it's like you just are never going to get like a high score there.
That's right. That's right. And that's why long read sequencing is so exciting, because you get bigger puzzle pieces where basically the vast majority of your sequences get high mapping quality scores. All right, so here's a bigger example. A bunch of different records, well, in fact, 5. It's hard to make out, but I've highlighted in green. Sorry for those that are colorblind. Red and green is not the best combination here. But 5 different records, and you can see it's basically just reporting chromosome, position, cigar string, sequence, all this stuff. So this gives us all the information we need to count reads that align to a particular gene, to call ChIP-seq peaks, ATAC-seq peaks, to find genetic variation, everything. You know, how these algorithms work to do that, we'll talk about next week. But you can imagine, I think, if you've ever looked at UCSC Genome Browser or IGV, essentially what you're looking at with each of those bars that are aligned to the reference genome is this information. This sequence aligned to this position from here to here. In an IGV, it's showing where all the differences are between the sequence and the reference genome. All that information is packed into this format. Okay? Yeah?
So it looks like on the top one, the match comes from a different chromosome, if I'm reading that right. It's about like
Yes.
10% as a 4. That's like a red flag, right?
That is either evidence of a translocation or a mobile element insertion or junk.
Okay.
Most likely junk. Yeah. So you'd only trust the alternative that it's a translocation. So, one end is aligning to chromosome 1, the other end is aligning to chromosome 4. You'd only believe that if there were lots of other reads supporting the same thing. Quick aside, in the early days of Illumina sequencing, you had to add these sequencing adapters, right? And those sequencing adapters are to tether the molecules to the flow cell. In the early days, that was done with ligase. And anytime you use ligase, you have the potential of actually stitching together two different molecules that belong on two different chromosomes, so you make a chimera. And then those get put on the sequencer, so you could get things like this. So that was especially pernicious if you're looking for things like mobile element insertions or structural variants, which was my entire postdoc. So I spent like the first year of my postdoc trying to figure out how to like detect chimeras and figure out what was real rearrangement versus artifact. Nowadays we use transposases to slam in those adapters, so there's no ligase, so chimeras are basically gone, which is really nice. Right, so last time we talked about edit distance, how many changes must be made to two different sequences to make them identical. We walked through these silly examples and talked about how there's different alternatives with different edit distances and figuring out the optimal alignment. And I emphasize that it's optimal. It's not. There is no correct alignment. There's an optimal alignment given the scoring scheme. Because if you remember that matrix that we filled out with the Smith-Waterman algorithm, you can imagine that the choice of path that you take in that traceback could change if the scoring scheme were different. So it's a reproducible optimal alignment given a scoring scheme. Doesn't mean it's the correct alignment. Okay, so in the BAM format, or SAM and CRAM and BAM, they're all the same, just different flavors of the same information. There's a field called a Cigar string. Could someone Google for me, so I don't have to do this, Google Cigar, SAM, and what the acronym stands for? I always forget. I know it has the word idiosyncratic in it, I believe, for reasons that are perhaps idiosyncratic. So that Cigar string is actually what's capturing the edit distance and the exact nucleotide by nucleotide differences between the sequence and the reference genome. Okay, so if any of you have ever had any computer science or know anything about compression, there's this concept called run length encoding. For instance, if you had 50 A's in a sequence that matched 50 consecutive A's in the reference genome, there's two ways of conveying that information. Write out 50 A's, which requires 50 bytes, or write out 50A, which requires 3 bytes. Basically saying there's a run of length 50 where the match is A. That's run length encoding. So you're compressing that long sequence into something small. Cigar string is essentially run length encoding. And the codes are things like match, a deletion with respect to the reference, an insertion with respect to the reference, denoted by M, D, and I, and N if there's a splice in the alignment. So RNA-seq data has the Cigar strings have a lot of N's in them to represent exon-exon, the split exon-exon boundaries in the cDNA. There's S and H for clipping. So remember in the last lecture when we did that dynamic programming, the local alignment, it removed a couple of the nucleotides at the beginning and the end because they didn't match. Those would be clipped nucleotides. So essentially, I think we clipped off the first nucleotide in that matrix. So you can imagine that the Cigar string would start with one S for the first nucleotide is soft clipped. Okay. And I actually forget what padding is used for, and it almost is never used. So let's walk through a couple of examples. I think this will make it clear. So imagine that BWA has taken this sequence, I call it experimental, this is just from the FASTQ file. It has found a location in the reference genome, and it has aligned it to the reference genome like this. Okay, so you can kind of squint to see what the edit distance is. So how is this encoded? First, the cigar string would start out with 4M. There's a run of 4 matches. Okay. Then, what's the guess for the, you can skip ahead, but don't. What's the guess for what the next encoding would be? 1D. There's a deletion with respect to the reference genome. Then 2 matches. Then 2 insertions. Then 7 matches. Hold on a second, what's going on there? So this is where it gets a little confusing. 7M means it's a block of sequences that don't have deletions, insertions, or gaps. It doesn't mean that there aren't mismatches therein. Okay? Which is a problem, right? Because that doesn't actually represent the total edit distance. That's where that tag, that optional tag field comes in. This would be the cigar string. Well, let me show you what the resulting cigar string would be. Let me table this discrepancy here for just a second. These last 2 nucleotides are not part of the alignment. It's a local sequence alignment. And we learned last time that a local sequence alignment cannot start nor end with mismatches. So these 2 nucleotides are not part of the alignment, so they're clipped. They're soft clipped. Okay? So the resulting cigar string would be, what we'd see in the SAM file would be 4M1D, 2M, 2I, 7M, 2S. Okay? In this case, the goal of the cigar string is to actually like encode all of this nucleotide by nucleotide information in a way that's more succinct than actually just writing out this alignment. In this case, it's pretty close. It's almost equivalent, the amount of characters that are required to encode this versus the actual alignment. But when you get out in 150 nucleotide sequences where the error rate is very low, usually you can just, what you'll see in an actual BAM file is often just like 151M. Everything's identical. So that's a lot, that's 4 characters rather than 151 characters. So there is a savings typically. Okay, so this mismatch, which is not reflected in this 7 match cigar encoding, is captured in those optional tags. It'll tell you at position, whatever this is, the reference was a C, your sequence was a T. And it's those tags that things like GATK and freebase and variant callers use to detect that there was actually a difference between the aligners. I walked through all that because that's sort of the way things were done till about 3 years ago. Now there's a new way of encoding this, which is better, which introduces instead of M, now we've got 2 different operations, an equal sign and an X sign. So equals is used when there's an exact match. So this is saying there's an exact match of length 4. We've got the same deletion, we've got an exact match of 2, we've got 2 insertions, we've got an exact match of 3, but now, previously this was 4M representing this block right here. Now it's 3 equal 1X. The X represents that there's actually a mismatch here. So now all the information you need is in one spot. It's all in the cigar string. That's the standard now, which I think, even though it's a little trickier to read maybe, it's more intuitive as to how this cigar string connects directly back to the sequence alignment that BWA produced using dynamic programming. Yeah? Okay. All right. So now we're getting into probably the hardest thing for me to explain in an intelligible way. So there's this flag column that is integers. For each sequence, each entry in the BAM file, there's an integer value. And that integer value is the representation of something called bit packing. Does people know what a bit is? 0 and 1, right? Computers use bits. The number, oh, it's great that we have a whiteboard, so I can actually do this justice. Before I move on to the next slide, let's just walk through some basics. Can people in the back see? I guess you can see the lower part, right? Okay, so if I write the number 4, this is in base 10, right? So in base 2, the equivalent of that, we start, let's just move backwards here. This is 2 to the 0, 2 to the 1, 2 to the 2, 2 to the 3. And by analogy, in base 10, it's the same thing. You got your 1's column, your 10's column, etc. Right? So if I wanted to represent the number 4 in binary, I just turn on this bit, because 2 squared is 4. If I want to represent the number 5, what would I do? Yeah, I'd flip this. Flip this one. Right? If I want to represent the number 7, I'd flip this one on. If I want to represent the number 8, I'd flip all those other ones to 0, back to 0. Right? Because 2 to the 3rd is 8. Okay? So the flag column, and so each of these can be thought of as flags. Each of these bits are answering a true or false question. So imagine that there's 4 bits that are being used here. Maybe I use this first bit to represent one question, like am I human or not? Yes or no. Am I an adult or not? Arguably no. Am I whatever. All these questions. You could encode the answers to all those questions into one relatively small number because we can use bits that only have 2 values. So that's the same idea that is being used here. Oh boy, let's see. Okay, so we've got all these different bits here. So this is the same as what I just drew on the board. This first bit, if true, if set to 1, means that the read that you're looking at came from a paired end sequencing molecule. If that bit is set to 0, that means you're dealing with single end sequencing. If it's set to 1, it's paired end sequencing. Okay. This second bit answers the question, is the read mapped in a proper pair? Proper pair means that the leftmost end of the aligned pair is on the forward strand and the rightmost end is on the negative strand. And if you think through the DNA gymnastics of your sequencing 5' to 3', that's the way it has to work out. So a proper pair is they're within the expected distance apart and the ends are pointing at one another. Okay? So that's what that second bit is for. The third bit, whose base 10 value is 4. This first one, the base 10 value is 1, 2. Now we're up at 4. And this is all powers of 2 because we're moving on different bits here. 2 to the 0, 2 to the 1, 2 to the 2, 2 to the 3, 4, 5, 6, etc. This one says, is the query mapped or unmapped? So this is one thing we haven't talked about yet. You could have a situation where BWA tries as much as it can to find a home for that sequence in the reference genome you gave it, but it can't. So that's indicative of 2 possible things, well, at least 2. One, that sequence is so crappy that it can't find any k-mers in the reference genome that match it. Or that sequence is a result of contamination. You thought you were sequencing mouse DNA, but you actually sequenced a little bit of your DNA too. Or the bacteria that were on your gloves or something like that. Okay? So it tells you yes or no, it's unmapped. What about its mate, the other 5' end? Is it mapped or unmapped? What about the strand? If it's 0, I think somewhat counterintuitively, if this bit is set to false, that means it's aligned on the forward strand, the reference genome strand. If it's true, that means it's on the reverse strand. So it's answering the question, is the query on the reverse strand? What's the strand of the mate? Is it if it's true, is the mate on the reverse strand? So the strand, these 2 bits are used to answer whether or not the read is in a proper pair, whether those 2 ends are pointing at one another. Okay, and so on and so forth. There's a lot of different questions that are done here. So that are answered in this one integer. So how do we end up going from the answers to all these questions to a number like 97? Let's see. I'm going to propose one way. 64 plus 32 is 96, plus 1 is 97. So 97 means that the query is in the first read in the pair. We've got a strand, the strand of the query's mate is reverse, and the read originated from a paired end sequencing molecule. So you basically look at the values of each of these bits. They have an associated base 10 value. You sum all those values up, and that's the integer value that goes into the flag column. Okay? So here's an example. If 2 to the 6th is set, that is 64. 2 to the 5th is set, that's 32. 2 to the 1st, so you end up with 99. All right? So you do not need to be developed into a savant where you can actually look at these scores and know what all the bits are. This is just to help you know what the heck is going on and how, when you go to IGV, it knows how to draw the alignment in this direction or this direction, or to color it such that it's mapped or unmapped, or to convey its whether or not the other end is on another chromosome, that kind of stuff. It's all embedded or bit packed into this one integer. Okay, you can forget about this forever if you want. If you need to know about this, just refer back to these slides and you can see how it works. Okay, so we've talked about SAM format. If your insidious goal was to use as much disk space as possible and like ruin your lab's server, store everything in SAM format. Your PI will love you. You'll have to be drawing hard drives all the time. So the original version of this format was text only, and that's because the throughput of the technologies actually weren't all that high. I mean, they were high relative to ancient technologies, but you were getting like 10 million reads, and you got a 10 million line text file, big deal. When it jumps by 1, 2, and even 3 orders of magnitude, and you're storing everything in text, hilariously bad things can happen to your hard drives. You just like one whole genome sequencing run. When I was a postdoc, my PI actually spent like a big chunk of his startup actually buying disk space so that we could store all the data from all the mouse lab strains that we were sequencing. So one way to get around that is to convert these text files, SAM format, which is super easy for a human to read, into its binary equivalent, hence the B in BAM, the binary alignment and mapping format. So basically that compresses, it basically recognizes the redundancy in that text file, eliminates that redundancy and compresses all the data. You've probably used a compress tool on your computer or gzip, something like that, same idea. All the information's there, it's just compressed. So this command that uses something called Samtools, also written by Hung Lee, this is basically saying the input format is SAM, capital S. I want you to emit BAM format, lowercase b. Why this isn't capital B, yeah, who knows. This takes a SAM file as input and outputs a BAM file. Okay, and what you'll see typically is that's like this file is like 10% the size of the original file. There's a new format that we're not talking about today that is also supported by Samtools. It's called CRAM. It's even tighter compression. now you're getting down to maybe even like 1% or 1 to 5% of the original file size. And if your lab is doing lots of sequencing and storing lots of data, it's pretty nice. And actually, for some of these, they get so small that you can actually put them on your laptop, view them on IGV on your own laptop, which is quite nice. Okay, so part of the homework is actually going to be to use some of these tools. So I'm not going to go into too much detail, but this view command is mainly used to convert, to actually show the contents of a SAM, BAM, or CRAM file, but also to convert between formats if you need to. So Samtools has a bunch of different options. Most of them really are not used all that often. The main ones are to index a SAM or BAM file. That's required so something like IGV can actually load it up and view it. And we talked about view. This is basically SAM to BAM to CRAM conversion. Okay, there's a lot of other options, but this is really, these other options are mainly used to fix minor mistakes that are in the format. There's a bunch of other ones. You can combine lots of different SAM files. There's actually a variant caller in Samtools called mpilup. It's not really terribly widely used. You can sort alignment files. So this is important. This is a critical step. A FASTQ file, those DNA sequences in the FASTQ file are in completely random order. I mean, you've taken 1 million cells, you've lysed them, you've put transposase in there, you've put adapters on all the molecules, and then that gets put in a tube. You then pipette that onto a flow cell, and those molecules are just scattered randomly across the flow cell. The sequences that you get in your FASTQ file are basically I think they start like at the upper left. Say they start at the upper left of the flow cell and are just reading out sequences in order on the flow cell, but essentially with respect to the reference genome, they're all in random order. So one of the first things we do with a SAM file is actually sort it. So the sequences for chromosome, the left end of chromosome 1 are first, then to the end of chromosome 1, then we get all the sequences from the beginning of chromosome 2 to the end, and so on and so forth. So the data are all organized. And then once they're organized like that with the sort command, we can index that BAM file so that IGV and other tools know, all right, you typed in the gene ATM, it's on whatever chromosome from here to here. It knows how to go into the BAM file and yank out the alignments that are germane to that locus, right? So to index them that way, the data has to be sorted by chromosome and position. That's what the Samtools sort command does. Okay. There's a couple different ways for installing Samtools. I'm going to update the slide. This is actually a bit outdated. They've changed it. What I'm going to do for you for the homework is install Samtools onto Malibu ourselves. But what I'd like to do maybe later in the class is have a way to just go through the installation of all the software that you will probably need when you do this work in your own lab. I will also update this slide to have the more relevant instructions. So now Samtools depends upon something called HTS-lib. You have to install that first, and then you can install Samtools. That's not reflected here. But there's a tutorial that I've developed a few years ago for working with Samtools, and for the homework, I would encourage you to do that. Samtools will actually be installed on Malibu, so you don't have to go through the installation step, but there's a bunch of examples of how to get data that's in BAM format and how to use these common commands to ultimately be able to display. Typically what you want to do is you do your sequencing experiment, you convert all the data, you do the alignment, you convert the data to BAM format, and you quickly want to be able to get that up into IGV or something and just look at the data. Did this experiment suck or was it good? And these steps really are the standard workflow for getting to that point. So that's one of the things we're going to have you do. So I'm just going to try to play around in 8 minutes with a BAM file using Samtools, just so I can get your feet wet. The other thing that I'd like you to do, you can do this at your leisure, but work with this tutorial that Obi and Malachi Griffith put together for using the integrated genomics viewer, IGV. This is how we actually look at high throughput sequencing data. It's not the greatest thing in the world. It crashes a lot. It's slow. It does some weird stuff, but it's the best thing we have. So you need to learn how to use it. All right. So quickly, I think I was stuck trying to install Samtools before class. Yep, it doesn't exist. So let's figure that out. I think I have it here. So if you go to Samtools, I just Googled Samtools. It took me to this GitHub page. It tells you about how to install. You got to run this stupid thing called auto header, then auto config, and then configure, then make install. I did that, and then it said, oh, no, that didn't work. And then I didn't read the fine print. It says, by default, this will build against an htslib source tree in .htslib. That is a cryptic way of saying you need to install htslib first. Okay, so how do we get htslib? If you go to the, this will take a few seconds for the internets to catch up. If you go to the GitHub/Samtools, there's also a repository for all the code related to something called htslib. HTS for high throughput sequencing, lib for library. This is a bunch of C code that John Marshall and Peter Danacek and Hung Lee and others have written. So all I need to do is say git clone, and that will download all that code into a directory. So there it is, htslib. I'm going to pretend I didn't attempt to install Samtools before. So I'm going to get rid of that. Pretend you haven't seen that command. Now I'm going to go to the Samtools page, but go to the Samtools repository. And I'm going to clone this repository. Okay, then I'm going to follow the directions. I need to go into the Samtools directory, and I'm literally just going to run these commands. Okay. These commands are used to basically figure out what type of computer Malibu is, and once it figures out what type of computer Malibu is, what kind of processor it has, what kind of compilers it has, we're getting into sort of software speak, it will know how to properly build the code that underlies Samtools into a binary that you can actually run on Malibu. This is cryptic stuff, I realize, but it's sort of something you have to do all the time to get software installed and running on your own system. Okay, so it failed. Why? Okay, so let's do this. It says either configure without curses. They don't mean swear words. I think I'm going to try this. That seemed to be better. Curses is something that's used to display crude graphics on the terminal. Malibu apparently doesn't have that installed, so configure complained about it. You've probably run the make command before. This is to compile all these .c files are source code written in the C programming language. There's a lot of code. If you go to the GitHub website and look through all the code that's been put into this, it's a monumental amount of effort. I think we owe the people that develop these tools a huge amount of gratitude because it's a ton of know-how. And now we found another error. So This thing called zlib, I think Simone at least ran into this. Zlib, at least the way that Samtools or CRAM format needs it, does not exist on Malibu. So this is an error that I cannot solve in the 2.5 minutes that we have. So I will try and install this zlib, and we'll try and finish up installing Samtools next time and go through this example. But rest assured that it'll be pre-installed for you for the homework, so you don't have to suffer through this. But I at least want to give you guys exposure to the process of trying to do this, because actually one of the hardest parts of using software in genomics, or really in any discipline, is just getting it installed, compiling it, and making it available. Just a reality. So lastly, in the 1 minute that I have, do this IGV tutorial. Try to do this Samtools tutorial. Each of them will take you maybe a half hour. That'll really set you up for knowing what you're doing with your own data in the lab. And then do this homework number 4. It's not due till the 18th. And I think I finally turned on this submission folder properly, so you don't have to worry about that. There's 30 seconds or a minute left. Any questions? Anyone have concerns or problems with existing homeworks or anything? No? Okay. Mark, one of the TAs, alerted me to the fact that one of the commands in homework 3 was broken. That's been fixed. He also alerted me to the fact that it looks like the Ensemble FTP site is down and you can't download those files. Are people struggling with that? It's back up? Okay, so they unplugged a computer or something and plugged it back in. Okay, well, if you have problems, let us know, but it sounds like everything's going well, so see you Tuesday. Yeah. I think today. I think it was I think that's what I said in class. I think there was a discrepancy between what was on the slide and what I said.